rather surprisingly got admitted to St Mary's Hospital for training, because I thought that was for the aristocracy, but obviously he managed to slip in there. And he was a classmate of Alexander Fleming, and we're in the building named after Alexander Fleming today. So I think there's a very interesting association there. I'd like to highlight that he was an assistant surgeon at the Royal Free Hospital, because obviously that's my clinical base, and it therefore seems entirely appropriate that for being an assistant at the Royal Free Hospital to being Professor of Surgery at St Mary's. That's a natural progression in surgery. So any trainees that want to come and work with me at the Royal Free Hospital, that's what's going to happen to you. Just 6 months and then you get a chair in surgery. Interesting about when we look back on what people did and what they achieved in their time. And it is interesting that his main hypothesis was that cancers were virus related. And although we might laugh at that off and on, it actually has panned out to be true for quite a lot of common cancers. So clearly his thoughts and his visions and his collaborations with his pathologist, which I think was very strong, did actually lead him up the right street in many ways. He obviously was a collaborator, and I think that one of the things and the themes that will come out of my talk today is the importance for all you guys who are trainees or even recently appointed consultants to think about collaboration for your development in the future. There are challenges, and one way to get around these challenges for the future is to collaborate and get good friends and supportive friends for taking your practice forward. And I think that that is a very, very important thing, both in clinical practice and in research. The other thing that's interesting is that this Pannett was a bit of a multitasker, which obviously I don't approve of, because men can't multitask. But not only was he a surgeon, but he was an anaesthetist and a pathologist. So he was really covering all bases, I would have thought. And what about the work that he was doing? Well, again, we have become so subspecialist, haven't we? We can't go outside our field, or there'll be somebody tapping on the shoulder. But back in his day, it was entirely reasonable to be both an upper GI and lower GI surgeon. And as you can see, he was publishing in high-quality publications. He was publishing in the Lancet his clinical work related to bowel cancer, which is really quite formidable. So what about my own speciality? I'm an HPV and liver transplant surgeon, and I thought I would just have one slide about some of the dates of what's been important over my career in training. I can assure you I wasn't around for the beginning, but it's kind of interesting to look back at the first liver resection being in 1888. And the cynics in the room might say, well, not many things have changed. The patient went back for a washout for bleeding the next day. But the patient did survive, and if you look away back at 1899, there was a publication of 70 patients having liver surgery with a mortality of 15%. So for 100 years ago, really quite exceptional results. And this is the importance of people having documented outcomes. And we heard earlier today that the lack of documented outcomes is one of the concerns regarding the developments in AI and its application to medicine. Other important things that happened in my field, transplantation, 1967, doesn't seem that long ago. I started in 85 transplanting, live donor transplantation at the end of the 80s, and then laparoscopic liver section in 1992, so really not that long ago that laparoscopic liver section commenced. And then we're in the throes of robotics. Again, a vacuous area from the point of view of evidence base, but it's being done frequently and in large centres. So my talk is image guidance for liver surgery, the next thing on that list. So what about augmented reality in general? So for those of you that don't have a particular interest, we know that for augmented reality often relates to just technology overlays of virtual images onto the real world. Tim Cook, who as you can see was having a great time at one of his recent meetings, felt that augmented reality will change everything. And I'm here perhaps to put more of a balanced perspective on where I think augmented reality might go in surgery. For most of the surgical applications, as I say, it's often a combination of pre-operative information with information obtained by the surgeon at the time of the procedure, or the equivalent of X-ray vision to view anatomy not typically exposed. And you'll see the examples that I'll show you shortly. When you look at predictions, it does seem as if it's predicted to be a huge impact on healthcare. And you can see my rather outdated figure here is that the immune-guided surgery market might be worth about £3 billion per year. So it's big business, but I think there is big potential, and I think we have to ask ourselves whether or not this potential is or is not being delivered. And if it's not, then why not? My own personal vision, I like visions, but usually my visions are pretty simple and straightforward. But my vision for this program of work was that we could have all patients that require liver surgery being able to benefit from laparoscopic or keyhole surgery. So, quite an easy throwaway line, but did it really mean anything? Well, if we're gathering information for a program of research work, we've got to try and say, well, what are the component parts? And as you know, we start off with component parts of who's going to benefit. And right at this day and age, unless the top of your application shows the patient benefit, I can guarantee that your application for research money will get absolutely nowhere. Then there's a question for the NHS. The NHS has no money, so if the second line in your application is that the patients will benefit and the NHS will save money, you're getting into a possibly useful grant application. And then we think about the big picture of society, and usually that means can you translate that across the UK, or can you translate that globally? Because that is big impact, and healthcare wants big impact. So what's the current situation about laparoscopic or keyhole surgery for the liver in the UK? Well, we got some information about that, and about 5 to 10% of all liver surgeries are being carried out laparoscopically in the UK. Some centres more, some centres less. And we have to ask ourselves, well, given that it's clearly of benefit to many, many aspects of surgical practice, why has that not been translated to liver surgery? I know most of you in the audience will say, well, that's because HBB surgeons are not any good at laparoscopic surgery, and you might be right. But there may be other reasons. It might be because the kind of operations we do are quite complex. We like to pretend they are, and that's why we've developed a speciality. So we think that it's a complex operation, and because it's complex and we can't palpate cancers, and we have difficulty visualizing them, even with the tools that we have intraoperatively, that we don't want to do things laparoscopically. And then most surgeons are most concerned of all about getting into major problems where they're faced with a mortality risk from a misadventure laparoscopically. So I think those are the drivers for why we haven't implemented laparoscopic surgery as much for liver surgery as it is in other areas. So our solution, which I'm going to discuss, was to get experts together to look at whether or not we could improve the quality of information we have for complex laparoscopic liver surgery. And we had, or I had collaborated for many, many years with a professor in image guidance surgery, or image guidance, he's a computer engineer, that's Dave Hawkes. And we got together a group of individuals that wanted to try and do something about this problem. We went to the Wellcome Trust, and they supported this initiative to try and transform laparoscopic liver surgery. And the solution that we went to them with was this. The idea was that we would develop a system for individualized patient care at the time of laparoscopic surgery based on taking their preoperative scans, and for those of you that are not very familiar, this is the big cancers here, this is the liver, and this is somebody that would be having a right or extended right hepatectomy. We would then, for that individual patient, we would make a 3D model, and we were using Mavis, who, as you know, have got a big foothold or footprint on these three-dimensional modelling. And using that three-dimensional model, at the time that that patient came to surgery, we had a setup where the laparoscope we were using for the surgery was tracked using a tracking system here by this tracking ultrasound in the operating theatre. And that was the premise, and that's what we wanted to achieve. There were many, many challenges to that, and this is just a short list of some of the problems. So what we were trying to do, we were trying to map a model from the preoperative model onto something at the time of surgery. The model had to fit. So if the model wasn't in the right place or it wasn't in the right orientation, then the model would be useless. So we had to have calibration of the instrumentation. We had to be able to track the model so that as the picture at the time of laparoscopy moved, the model moved with it so that we knew where we were. We had the issue of the fact that this would have to go into theatres, and therefore everything would have to be sterile, it would have to be mobile. There was no point in having something that took up a room this size, and we then wanted to take it into theatre. We had to have a system that was reliable, and we've been discussing earlier this morning this fascinating question of when it goes wrong, is it the surgeon's fault? And of course the answer was yes. You're using this, it's not using you, so you have to take responsibility. And then there's the question of, even if we can produce the model and overlay the model on the liver, as we do the operation, the liver will change shape, it will deform, and then we have to re-register the model. So re-registration was going to be key. And not only was it re-registration with the surgery, but even pneumoperitoneum changes the shape of your liver, and breathing with ventilation changes the shape. So there was a lot of challenges there. So we started off with simple things, the technology, the equipment, can it work, can you put the component parts together? So in a lab-based system we used a phantom of the liver. We then got the system that we were going to work up for theatre. This is the ultrasound tracking equipment. This is the tracker that goes on the end of the laparoscope that's going to go into the patient. And we did some development work to see how accurately we could track the liver with the model being superimposed on the liver. And there's many systems, as I'm sure you know better than I do, for tracking and getting a model to overlay with real life. Unfortunately, most of these tracking systems have not been for deformable structures. So one of the starting points was this difficulty of getting the model to overlay with the liver, and we started working with this. So at the time of the laparoscopic procedure, we would use this checkboard in different angles, and we would use that to calibrate the system and to get calibration for an individual procedure. We did try other things. We looked at these systems for getting more detailed calibration. The more detailed the pattern that you can reproduce, the easier it is for you to track something in real life. And we tried these patternings on the end of the laparoscope so that we could use that to facilitate tracking. And that works very well, but to take that to a clinical product is not that easy, because you then modify the laparoscope, and therefore there becomes the question of whether or not you're taking responsibility for modifying another manufacturing equipment. And we went to some very basic ideas of whether or not we could calibrate the laparoscope just using a simple cross configuration. And it was actually quite accurate. You could do that. But it wasn't accurate enough, even in a phantom model, to take to clinical practice. One of the other things that we tried was to look at the question of whether or not we could track using a stereo laparoscope, because you could actually use the 2 ports of the stereo vision to actually identify the site that you were on the liver. Unfortunately, this required you to buy a stereo laparoscope. It required you to operate with 3D glasses on, and I personally found it really quite difficult to do so. But we did some development work that came through to clinical practice to develop whether or not a 3D system could be used for tracking. One of the problems with the liver is that I'm sure most of you here think that it's pretty amorphous and rather boring. Liver surgeons are very excited about the liver because we look at it and we think of all the structures inside, but I'm sure most of you look at that and think, well, that's just a flat sheet of meat. And that's one of the problems, that actually, if you try and track the surface of the liver, and the liver is smooth, there's not any actual bits to identify. So surface tracking on the liver is an incredibly difficult thing to do, certainly for a healthy liver, and even with this, which is a mildly cirrhotic liver, we couldn't manage to do accurate surface tracking. And our engineers came up with lots of different solutions. This was one of them, where we would look at having small areas of the liver over the contour of the liver, and we would use the tracking by taking those small patches, put them all together, and then transpose that on the preoperative model so that we could then move it. That was one of the most accurate things in the lab, and we did try and do that in clinical practice. So eventually we got a system that was working where we could track the laparoscope, and we could use it to track and then superimpose a model on a liver in the lab. And we had an accuracy of 3 millimeters in that. So if we were tracking the edge of the liver, it was plus or minus 3 millimeters. If we were tracking the vessels inside the model, and we did produce a model that actually had an internal structure that we could track, the accuracy was very good, down to 3 millimeters. At which point we decided that we would take it the next step, and we would produce a porcine model on which to evaluate the equipment. Now, there wasn't really any porcine models about, and what we really wanted to do was have a porcine model with a liver cancer that we would do laparoscopic liver surgery on. So, wishful thinking, you could spend a good few PhDs trying to produce such a model, and we decided that that really wasn't worth the effort. So what we decided was that we would produce lesions in the liver of the live pig. We would then get CT imaging of the liver in the pigs when they're alive. We would then produce the 3D model of the pig livers, which was again done by ERCAD. And then after we had the 3D model, we'd go back and we would apply laparoscopic image-guided surgery to resect the liver tumours. So, as you can imagine, it was an awful lot more difficult than we thought, which is what you find in every bit of research. Even finding the pig liver and separating it from the pig spleen is not that easy. The pig has a huge big spleen and it seems to get all over the place. So we spent the first few weeks just trying to work out on ultrasound exactly where the different sites were. We then, under ultrasound guidance, we used the microwave ablation probe to produce some focal lesions in the liver. So these were KID-ON or pseudo tumours, and we were using this purely to see whether or not the system could recognize them and could produce a model with these pseudo tumours. And this is a little tiny anaesthetized in the CT scanner. If you think that it might have cost us a lot of money to do this work, it was about £8,000 every time we did a CT scan in a pig, so you can just imagine the cost of this project. So, what did we do after that? Well, so we got the CT scan of the pig liver that had the microwave ablation zones in the liver. This is a typical picture, so these are the ablation zones here at the front. We produced a model, and this is the kind of 3D model we had of the pig liver with ablation zones. As you can see, this was me trying to work out the pig's liver anatomy, because it's very different from the human. So I had to go on a bit of a refresher course to work out exactly what was what with the internal structure of the pig anatomy. But in this work, we did actually find that we could align all the equipment. The alignment probes at the back here, there will be an ultrasound machine that is finding the position of those at the other side of the theatre there. And this is just one example of where we got to in that porcine work. I think the video is actually running, although it's about what's at the same speed as myself. So you can see here we're tracking these markers on the end of the laparoscope. You have to take my word that that's a pig liver. You can take my word that that's a pig spleen. So we're running the tracked laparoscope across the liver, and this is the 3D overlaid model. So you can see the ultrasound here. This is the marker of the vessels in the liver, and you can see that as an overlay, and you can see this is just confirming it on a cross-sectional ultrasound. So, we managed to have a system that was working. We managed to have overlay. It wasn't in real time because it was actually taking us longer to produce the models and the overlay than we could in real time. But certainly with reconstructions post-hoc, we could have some very good images and a high level of accuracy. It wasn't as good as it was in the lab, nothing ever is, but we had an accuracy of about 10 millimeters, and that included the errors for initialization of the equipment, for the surface reconstruction, for the registration, and for identifying any particular sites that you wanted to identify and work out the error. And it included deformation with breathing. So these experiments slightly overlapped, although in research you like to always pretend that things go from one successful experiment to another. In real life it never ever works like that. So we had actually started doing some clinical work at this stage, and we were going from clinical practice back to developments in the porcine model, back to developments in the lab, and to and fro, which perhaps is the right way of doing developmental research. Eventually we did get into a clinical study. We consented the patients for the collection of their preoperative imaging, the production of a model, and the production of an overlay either in real time or outside the body. So they were being consented for the model being produced. It was specific in the consent that I did not change the operation based on the information. Again, something that was discussed this morning. Who is responsible for the decision making? Well, our ethics board felt that it was inappropriate with a developmental device for any change in the practice to be done as a result of the information. So they would not allow us to actually look at the production of the model at the time we did the operation. So we had to evaluate that afterwards. But in this body of work we found, not surprisingly, huge challenges. It was actually it took quite a long time to set up in theatres, 3/4 of an hour, maybe an hour. We had some reliability issues. We really couldn't run the system without a full-time PhD postdoc in computer engineering, and they're very expensive. The interface was produced by a computer engineer, so I couldn't even switch the machine on, never mind work it. It was very complicated. And our accuracy in clinical studies was worse than it had been in the porcine studies. So there was certainly more variability. It was interesting. We had some very accurate procedures and some completely inaccurate procedures. There was a huge amount of variability. So that work and some of the other development work was done by Crispin, who was one of my PhD students, and he's just putting that data together. And there was a lot of development work because all of these things had been completely untried and tested. And as you can see, over that course of a few years, there was lots of basic science publications related to the technology and its application. So at the end of the programme, we then had to sort of take stock and say, well, what next? Is this of benefit? Have we shown patient benefit? No. Is there potential benefit? Well, if the system works, we can increase the number of patients having laparoscopic surgery, which would have some benefit. But interestingly enough, the UK and international data would suggest that the cost saving of doing things laparoscopically is not enormous, but significant. And then there's the question of, well, are we doing it for device development to get into this market? Are we doing it for IP because there's a lot of new algorithms being developed? Or are we doing it for other issues, such as perhaps not the NHS costs or patient benefit in hospital, but, for example, the importance of recovery period, because patients take a long time to recover after major open liver surgery. There was some competitors in the block, really only one that got to a commercial system that you probably know about, Cassination. They were trying to sell an open surgery system. There was a different methodology for registration, which was manual and based on drawing manually the outline of the liver, which we felt was not ever going to get to a product for laparoscopic surgery. So we felt that we were on the right stream. We had developed it to a clinical system. We had done some porcine model. We hadn't done any safety aspects as yet, but we hadn't applied it to change clinical decisions. But we had got a system that was manageable and usable and could actually give us the information that we felt was potentially important. So we felt that it really just required more development. We had originally hoped that we would do all the development work within the first 5 years, but it was quite apparent that although we had overcome a vast number of hurdles, we hadn't overcome them all. So at that stage, Matt took over the technical lead for the project, and he and I and a very nice PPI representative went and did a pitch to NIHR I4I, which, as those of you in the room that are looking for grant applications will know, that this is one of the organisations that are interested in developmental work, particularly device development. They liked the idea. They thought it was at the right stage for them, so they actually supported our development. So we had this initial body of work done by the Wellcome Trust, or supported by the Wellcome Trust. We had developed the clinical system. We had a proof of concept. We then wanted to move into a project that was going to look at safety, in particular, single-site utilization, and improvement of the system from the point of view of usability, and get surgeons that could actually use it. That wouldn't actually take us to the market, or certainly our feeling was that that wouldn't take us to the market, that we would require some additional evidence of benefit outside a specialist centre that had a vested interest in developing it. But this was our pattern of development that we took to NIHR for I4I, and they supported it, and we're currently just started an award of almost £1.5 million to develop this further for clinical practice. And these are the challenges and the goals that we want to achieve. We're still working on better calibration, particularly making it a calibration system that the nurses can set up rather than myself or our computer engineers. We are doing more work from the point of view of the 3D model, and we want to implement both planning for the surgery as well as the overlays, so to use the same model for both planning and the intervention itself, and join those two up. We're then developing some further empiric systems for improving registration, which has improved throughout the course of the work, but is still not as good as we would like. So we want to improve the overall registration accuracy. The first thing we had to start with was actually being allowed to use the system and look at the information during the operation. So we now have ethics to do that. It is ethics only to allow us to do it within the development site. So we are restricted, and that we can't, but we can now use the information obtained at the time. We're doing further work with new calibration methods, and some of the new calibration methods that we've produced not only have been published, but have accuracies beyond anything in the literature. So it's now the question of transporting those into the new smart surgery system. So it's become very much an iterative process. A new bit of work is done, it's then integrated within the system to try and improve the accuracy parameters. And we've also looked at the alternative of using different tracking methodologies. We've worked for many years on ultrasound tracking, and you can use electromagnetic tracking. And we've got a collaboration with a device manufacturer so that we've got a scope that actually has an electromagnetic sensor at the tip of the scope. And the plan with that is obviously to use that to develop greater accuracy in the guidance of the model in real time. Apart from those development works, there's all the question of product development, because it's now got down to manufacturer. To begin with, it was all done in our lab, it was all glued together, we had sticky tape, all the things that computer engineers love, but now it's the real world and it has to be a proper device. So we've had a lot of advice on how to manufacture some of the bits and how we can make them disposable. And we've also gone in reverse in a few steps. We've got some new technology that can produce a global snapshot of the liver. So rather than taking it in small segments, we're working on taking the global picture of the liver and using that for registration purposes. And much as you would perhaps, it's counterintuitive, it's actually shown accuracy that is much better, again, than the other systems that we've used in the lab. And we have applied it also to some archival human data, but not in theatre. So lastly, on the smart surgery project, and I'm going to just for a few minutes touch on some of the other work that we're involved with at the moment. But lastly, from smart surgery, we've had engagement with the public, and it's a key criteria at this stage of development to engage. This is Mustafa, who's one of my PhD students from Egypt, who's trying to explain to somebody how the system works, I think. And this is the engineering team, because this is absolutely key that you have the right component parts. And this is the engineering team who actually nicely won an award for this open day. So lastly, I'd just like to leave you with a few things that we're working on that are already funded. I can't tell you the things we're working on that aren't funded, but I can tell you some of the things that we've actually got up and running. We are obviously looking at image-guided surgery for other organs. We are working with robotics to implement the system in the robotic system. We're applying it also to other areas, and I'll very briefly mention the other areas that I think are extremely exciting areas for development. This is one of our projects that's funded through Cancer Research UK, and this, apologies, it's gone away from liver now. This is for pancreatic malignancy, or actually pancreatic pre-malignancy. We've got a study running that Steve's PI of, looking at endoscopic ultrasound RF ablation of pre-malignancy. Now, the destruction of pre-malignant pancreatic lesions, as you all know, is an unbelievably important goal in society. One of the problems is that you don't actually quite know which bit of the lesions to ablate. And this project has been funded to again produce a 3D model of the pancreas with the lesion in it, and use that at the time of the endoscopic ablation to guide the ablation site to the area of pre-malignancy. So we've been doing some work on a phantom, we've done some work on tracking the laparoscope, and this project is getting towards clinical application. The other project I thought you might be interested in is this project, which is looking again at pancreatic cancer. It's looking at this IRE therapy, which you may know of. It's an electrolytic therapy for cancers, and it's mainly been used for locally advanced and unresectable cancers. As some of you may know, I'm trying to get a UK trial running on this because this has not really been adapted in the or adopted in the UK. The overall data would suggest that it probably doubles the life expectancy of people with locally advanced pancreatic cancer. But there are problems to it, and one of the problems to this technology is that you've got to put electrodes through the skin into the pancreatic cancer, and there is no way of guiding appropriate electrode positioning. So the concept behind this project is that we would again use our image guidance system that we developed for smart surgery, and we would use it to guide electrodes for ablation through IRE and pancreatic malignancy. So again, this project is running. We've been doing some experimental work in the lab. It hasn't yet got to clinical practice, although hopefully it might dovetail with a UK clinical trial related to IRE therapy for pancreatic cancer. And lastly, I'm going to leave you with this other concept. One of the threads that we've had all through our development work is, okay, we're developing a system that might be able to tell us where the vessels are that we want to cut and divide under more controlled environment. We don't really have good enough feel for where we are in relationship to the cancers. So we're working on photoacoustic imaging. And photoacoustic imaging, as you know, is a way of determining the tissue characteristics even deep within the liver, because it's got several centimeters of penetration, unlike most optical modalities that can only penetrate a couple of millimeters onto the surface of the liver. So we've just been awarded a fairly large grant from Cancer Research UK to develop a new probe system for image-guided liver surgery. And the concept there is that we would use this in tandem with the system to determine not only where the cancers are, but that we're absolutely clear that we're well aware from the cancers when we're dividing the parenchyma. And lastly, I would say that some of the development work that we've been doing has all come together with the recognition of the Wellcome Trust, who have now funded us to have a VICE, which is our Wellcome Trust and EPSRC Centre for Surgical Innovation. They've funded it to a large amount, and this has spun out some of the developments I've been discussing. And I'd just like to thank all the people now that are within that bigger infrastructure that is developing very, very nicely to bring people together, physicians, basic engineers, along with PPI individuals, to develop some technologies, hopefully, that will transform the future. Thank you very much indeed.
Thank you, Professor, for demonstrating what is really quite an overwhelming body of work, going right back to the start and the field review. It's really nice to see Dan Stoyanov,